Welcome to our second lecture on smooth muscle. And now we're going to look at some of the characteristics of smooth muscle contraction and then compare single unit versus multi-unit. And you can print off worksheet 8.2 now. And to start things off on the right foot, we got a couple memes here. We got kids exploring ancient ruins. I remember there was still one of these outside of our science building up in Duluth. I thought that was pretty funny. It just got graffiti on it, and then I think somebody eventually hit it with their car. And then we got, why is dentistry important? Because even though he's missing an eyebrow, the first thing you notice is his smile. So that's some good marketing there. We'll start today by looking at the types of smooth muscle contraction, and then we'll move into the events that need to occur for relaxation of smooth muscle. To begin today, we're going to look at some contractile activity within smooth muscle. Now, smooth muscle can be grouped into two categories, and that's depending on that contractile activity or the state of contraction. And that is tonic or phasic smooth muscle. So let's start with the top. On this figure here, we can see we have this state of contraction here on our y-axis with fully relaxed at the bottom, fully contracted at the top, and then time on our x-axis. Now, tonic smooth muscle is either contracted or partially contracted at all times, and that's exhibiting muscle tone, hence tonic smooth muscle. And that's due to a relatively low membrane resting potential. And because of this, some of those surface membrane voltage-gated calcium channels we talked about at the end of last lecture, they're open at all times due to that lower membrane potential. So then calcium has a way into that cell to help maintain a contraction or partial contraction, or what we call tone. Tonic muscle won't show bursts of activity like we'll see with some other types of contractions. Instead, it's going to maintain that tone or a state of contraction. So two things here we're going to talk about. We can see here fully contracted, hopefully, are some sphincters. And then this period here would be the opening of that sphincter where we get full relaxation of that ring of smooth muscle. Maybe we're talking about a urethral sphincter here where we need contraction to prevent us from urinating, and then we'll get release. And then we can compare that to some partially contracted smooth muscle, which we spoke about before. We said the blood vessels, right? So the arterioles, for example, the smooth muscle in the walls of the arterioles is in a state of tonic smooth muscle contraction. That ongoing contraction squeezes down on that blood that flows through them and helps maintain blood pressure. In contrast then to our tonic smooth muscle, we have phasic smooth muscle. And phasic smooth muscle, it contracts in bursts. So this is triggered by action potentials, which lead to opening of voltage-gated calcium channels. So we get this influx of calcium, a rise in cytosolic calcium. And that leads to pronounced increases in contractile activity. So phasic smooth muscle commonly exhibit these rhythmic contractions that we see here. For example, we'll see these within the gastrointestinal tract. So for things like the stomach and the intestines, the smooth muscle there is abundant within those hollow walls of those organs, and that's going to move the contents through them. Just to reiterate some key points from the last lecture, with skeletal muscle, we would typically see a release of calcium from the sarcoplasmic reticulum, and then that calcium would bind to troponin and reposition the tropomyosin and allow the myosin head to bind to actin. With smooth muscle, those voltage-gated calcium channels will open, leading to an influx, mainly from the interstitial fluid or the extracellular fluid, into that cell. Once the calcium is in the cell, it's going to bind calmodulin. That calcium calmodulin complex activates the myosin light chain kinase, and that kinase is going to phosphorylate the myosin light chain, that little light green necklace around the neck of this myosin, okay? And then it can interact with the actin molecules in the thin filaments. We're now familiar with what leads to contraction of smooth muscle, but what stops it? It's a little more complicated than skeletal muscle relaxation. There's another added component we need to take into consideration, but it does have some things in common with skeletal muscle as well. So let's look at those. The first thing we need is the cessation of stimulation. So what this means is just remove the stimulus and we stop the action potential. Okay. If we stop that action potential, that'll stop the opening of these voltage-gated calcium channels. Okay, so we inhibit those. And then we still have the calcium within the cytosol of that cell, or the sarcoplasm. So we need to activate the calcium pump exchangers. You're familiar with these when we talked about skeletal muscle. We need to sequester that calcium again back into the sarcoplasmic reticulum, and this is the action of SERCA pumps. That's the sarcoendoplasmic reticulum transport ATPase, or SERCA. It's going to move those calcium ions from the cytoplasm of that cell into the sarcoplasmic reticulum. An additional step that is needed with smooth muscle is going to be removal of this phosphate group from the myosin light chain kinase. So recall that it's the addition of that phosphate group that permits the binding with the actin molecules. It allows this myosin head to bind actin. So while we needed the myosin light chain kinase to add this phosphate group, we're now going to need the myosin light chain phosphatase to remove that phosphate group from the myosin light chain. And as a little FYI here, muscle may not relax right away, smooth muscle at least, and that's due to that latch bridge mechanism that I mentioned earlier. So let's take a look at that and see why that is. The latch bridge mechanism is a product of the myosin heads having modifications that allow them to latch onto the actin of thin filaments. So they remain attached without using additional ATP. So it keeps the myosin head attached to the thin filament for this extended period of time. It forms a latch bridge. So we see this prolonged, maintained contraction or tension with the smooth muscle. This is one of the theories for how sphincters stay completely contracted without exhibiting fatigue. But the latch bridge mechanism isn't completely understood yet. And we'll now look at some defining characteristics of a smooth muscle contraction. We'll now review some of the common characteristics of smooth muscle contractions, and we'll begin with the longer latent period. And it's a longer latent period due to two reasons, and the first is it takes time to phosphorylate the myosin head, so to put that phosphate group on that myosin light chain. So here perhaps we have a better depiction. Here we have our calmodulin, that protein that binds the incoming calcium. We form this calcium calmodulin complex, which then activates this myosin light chain kinase. This myosin light chain kinase then uses ATP to phosphorylate the myosin light chain on the myosin head. And all these events take time. And the other reason is there is slow ATP ACE activity on the myosin head itself. So remember we have that myosin ATPase site where we split ATP into ADP and the phosphate group. Well, the splitting of that ATP can take time as well. In fact, all these events together take about half a second. And on a physiological time scale, that's actually a really long time. So we see that there can be a longer latent period for smooth muscle contraction or onset to contraction. Another characteristic of smooth muscle contractions are their long duration. On average, they last about 1 to 2 seconds. And one reason they're extended that we've mentioned before is that it takes time to remove calcium from the inside of that cell, removing the calcium from the cytosol. And we mentioned the SERCA pump as an example a few slides ago that sequesters the calcium back into the sarcoplasmic reticulum. But that reticulum, remember, is fairly limited within smooth muscle. So we also need to pump calcium back out into the extracellular fluid. So we have these calcium pumps in the membrane, and they're aptly named the plasma membrane calcium ATPase. And you'll remember these from last semester. We called them the PIMCA, the PMCA. And It's a transport protein in the plasma membrane of the cells, and it functions to remove the calcium. It puts it back out into the extracellular fluid. We also have these sodium-calcium exchangers within the membrane, and they help contribute to the removal of excess cytosolic calcium as well. And as previously mentioned, that long duration is also a product of the dephosphorylation of the myosin head by the myosin light chain phosphatase, as well as the locking of that myosin to the actin due to that latch bridge mechanism. At first, it may seem like these contraction characteristics are not ideal, but unlike skeletal muscle, we rarely need the rapid onset of contractions with smooth muscle. And these extended contractions are well suited for their functional requirements. So for example, it's important to maintain tone in the GI tract and vessels where we need slow contractions for peristalsis, for example, to move contents through the GI, or we need vascular tone to help propel our blood. An additional characteristic of a smooth muscle contraction is that those contractions are fatigue resistant. And that's because the energy requirements are pretty low compared to skeletal muscle. They're primarily fueled by aerobic respiration. And if you think back to the end of last semester when we talked about metabolism, that's one of the slowest sources of ATP, provided you have oxygen and a fuel source. The other reason is they can maintain contraction without added ATP, and that's because of that latch bridge mechanism we defined earlier. And that allows us to maintain that contraction or tone. And the last attribute of a smooth muscle contraction we'll talk about today is the broader length tension curve. So here we have the skeletal muscle length tension curve you'll recognize from last semester. And recall that the force of a muscle contraction for skeletal muscle is dependent upon its length at the time of stimulation, or the start of that contraction. So it shows maximum force at the optimal resting length, but we see a decreased force of contraction if it's shortened or if it's lengthened. And we don't see these limitations with smooth muscle. And one of the reasons is we lack Z discs. So here we can see our Z discs within our skeletal muscle. And we can only shorten that sarcomere so much before we run up against those Z discs. Well, we don't have those limitations with our smooth muscle contractile protein arrangement shown here. The other reason is we have myosin heads present in the center of the thick filaments with smooth muscle, while with skeletal muscle, remember, they're limited to the ends of those thick filaments. So what that allows for is powerful contractions, even if we're compressed or stretched. We can still have a forceful contraction. In fact, we can contract forcefully even when we're at 50% or 200% of our resting length. What does that characteristic of smooth muscle contraction allow for? Well, consider this scenario here. Say you've been holding your bladder for a very long time. Well, the more amount of urine within that bladder, the more that bladder is going to be distended, right? And therefore the smooth muscle within that urinary bladder wall. Well, the ability of smooth muscle to contract forcefully at different degrees of stretch is going to allow us to easily empty our bladder regardless of the amount of urine that it's been holding. Moving on, we'll compare multi-unit to single unit smooth muscle and talk about whether they are myogenic or neurogenic. When talking about multi-unit versus single unit smooth muscle, they're fairly simple to keep straight because they're kind of named accordingly. So as that name implies, a multi-unit smooth muscle is going to consist of discrete units that function independently of one another. And therefore they got to be separately stimulated by nerves in order to contract. And this is similar to skeletal muscle motor units. So we see here we have one motor unit. We have this incoming nerve going to these clustering of these smooth muscle cells, innervating them. And then let's say we have another nerve coming in here, and it innervates another cluster of smooth muscle cells. So this would be another unit. So we say that these are multi-unit. And those units have to be separately stimulated in order to contract. And we say that they are neurogenic. And when we say neurogenic, that means that the contraction occurs in response to stimulation by nerves. Now, the muscle cells in these structures, as we can see, have these neuromuscular junctions. Okay? And the degree of contraction is going to be due to how many of these motor units are activated, just like in skeletal muscle. But remember that skeletal muscle is voluntary. It's using motor neurons, while this is involuntary. We're talking about autonomic innervation here. So some examples of these would be in the walls of our large blood vessels and in the airways, large airways to the lungs. With multi-unit smooth muscle then, we see that there are few gap junctions between the individual smooth muscle cells. So we can't stimulate in one cell and then it'll spread through a gap junction to the next to the next. We instead have separate innervation by nerve fibers, and they have to be stimulated independently, those smooth muscle units. And this allows for finer control of different structures. And some of those are going to be the iris, for example, or the ciliary muscles that need that fine control to work properly. Now we'll turn to single unit smooth muscle. And most smooth muscle is single unit smooth muscle. And the term single unit smooth muscle comes from the fact that the muscle fibers that make up this type of muscle, they become excited and contract as a single unit, hence single unit smooth muscle. The muscle fibers in the single unit smooth muscle, they're linked by gap junctions. So when an action potential occurs anywhere within a sheet of smooth muscle, remember it's about 2 to 3 layers of these smooth muscle cells that make up a sheet, well, then that signal propagates via those gap junctions through all of those interconnected cells, and then they contract as a single coordinated unit. Okay. Another difference between single unit and multi-unit smooth muscle is the lack of a neuromuscular junction or motor end plate with these single unit smooth muscle cells. So we have a nerve fiber here, an autonomic neuron, and with a multi-unit, we would see that this would innervate this cell directly. Now, we don't have that with our single unit. Instead, we have these bumps here, or varicosities. They're just swellings along that autonomic neuron, and they contain neurotransmitter within some vesicles. Those can be released, and they're going to bind to receptors that are found all over the sarcolemma of this smooth muscle cell. So they're not directly innervated, but we get neurotransmitter release from these varicosities. And then that signal can spread via gap junctions to all the other smooth muscle cells. And because the signal spreads throughout that group of interconnected cells via the gap junctions, and they contract as a single unit, we say that they are a functional synctium. So syn means together, and then cyte, right? Like cyto means cell. So you can think of them as contracting as one cell all at once. And we see that this contraction is slow and energy efficient, we've mentioned before. So it's well suited for forming the walls of our distensible hollow organs, which is going to bring us back to the example that I keep bringing up of the uterine wall, the muscle there. So that's smooth muscle, and it acts as a functional synctium. And this is good because what we get is this slow, coordinated contraction of all those cells at once that push out the baby to deliver that baby. If we had independent, uncoordinated contractions, as we would with multi-unit smooth muscle, that wouldn't properly exert force to expel the baby. Let's compare multi-unit and single unit side by side. We'll start with multi-unit, and we'll see here we have this incoming autonomic nerve fiber, and it's going to nervate a few of these smooth muscle cells. So if we get signal here, or stimulation, we'll see contraction of that cluster of cells. Now, let's say we have another incoming nerve fiber, and it innervates a few of these cells. Well, we're going to need an independent stimulation of this nerve fiber if we want that cluster of smooth muscle cells to contract. right? And there are not gap junctions found between these clustering of cells. So we're just going to see independent contraction with a stimulation of a unit. We can compare that then to single unit, where if we get neurotransmitter release from vesicle and it binds a receptor, that signal is going to propagate through all the cells that are connected via gap junctions, and we're going to see that those muscles contract as a single unit. And the last thing we'll say about single unit smooth muscle is that it is myogenic. That prefix myo meaning muscle and genic like genesis. So we can say that genesis, or the origin of the signal for contraction, originates within the muscle tissue rather than from nerve impulses. So it's self-excitable. It doesn't need incoming nerve signals from autonomic fibers in order to contract. So let's look at this image here. So we'll see here we have this pacemaker cell. So it's going to have spontaneous action potential activity. It's going to spontaneously depolarize, what's being shown here. Okay? And that signal is going to spread via gap junctions to all the cells that it's connected to with this single unit smooth muscle. So we're going to spontaneous depolarization, and that's going to spread it and give us one unified contraction. Now, I know what you're thinking, because I just showed you this image on a previous slide with our single unit smooth muscle, where we had the autonomic nerve fiber and we're releasing neurotransmitter that's binding to receptors. And then they're moving through these gap junctions, so we get that whole contraction. Well, this is a key point. Although single unit smooth muscle is innervated by the autonomic nervous system, as we've shown here, the nerves do not initiate the contraction. Instead, they work to modify the rate or strength of that spontaneous contraction. And there are two kinds of spontaneous depolarizations, or those action potentials that occur without external stimulation that originate within that muscle tissue. And they are pacemaker potentials and slow wave potentials. So both these will initiate that muscle contraction, and then we can see modification by the autonomic nervous system, right? So maybe we'll get some acetylcholine or norepinephrine. The enteric nervous system, that nervous system is kind of associated with the GI tract, or maybe endocrine, hormones. We'll now look at the differences between pacemaker and slow wave potentials. The first of the two we'll talk about is the pacemaker potential. And this is where we see the membrane gradually depolarizing on its own due to some changes in ion channel permeability. When the membrane has depolarized and we hit threshold, we'll get an action potential. Then it's going to repolarize, and the membrane potential again will depolarize to threshold, giving us another action potential. And it's going to occur like this in a cyclical manner over and over. Now, the pacemaker cells, while they can initiate the action potentials, they cannot contract. There's only a few of all the cells within that muscle tissue that are non-contractile pacemaker cells. The rest are contractile cells, but the signal from the pacemaker cells spreads into those contractile cells, so we get that mass contraction. In contrast then to the regular rhythmic cyclical patterns of depolarization that we saw with pacemaker potential, we have slow wave potential. And it's unknown mechanism how we get these slow wave potentials, how they occur, but we see that they're alternating depolarizing and hyperpolarizing swings in potential. And they may or may not reach threshold, but when they do hit threshold, you get this burst of action potentials that will lead to contractile activity. These are only found within the digestive tract, and they seem to be associated with clusters of pacemaker cells. And the pacemaker cells send out the signal to contractile cells surrounding them. And they're going to spread through those contractile cells via gap junctions so that we get contractile activity. Now, the threshold, it may be more apt to be reached if we have an influence of neural factors. So if we have some autonomic input, or maybe there's other local factors like we just ingested a big meal and we need to kick off peristalsis. To finish today, we're going to look at how we can modify the rate or strength of a single unit smooth muscle cell contraction. So there's pacemaker cells that initiate the contraction, but then we can modify the rate or strength based on autonomic inputs. So we're going to take a look at that. We're going to look at parasympathetic and sympathetic inputs, specifically looking at acetylcholine and norepinephrine, and how we can change contraction. We're going to look at some cell signaling events, and specifically we're going to look at a smooth muscle cell within the intestines as an example. We'll start by drawing our contractile cell. Oops, way off the board here. Alright, so this is a single unit smooth muscle contractile cell. And we're going to look at the effects of some autonomic inputs. So parasympathetic innervation, so that'll be effect of acetylcholine, as well as sympathetic innervation. So that'll be norepinephrine. Now, to start off, we're going to have some type of spontaneous depolarizing activity. So we got these depolarizations, okay? And we know that those lead to opening of voltage-gated calcium channels. So I'll throw those here. All right, I'm going to abbreviate, so we're going to do V-gated Ca2 plus channels, and that's going to lead to an influx of calcium. Okay. Now, we also reviewed that calcium, upon entering the cell, it binds calmodulin and forms the calcium-calmodulin complex. Okay. And that goes on and activates our myosin light chain kinase. That myosin light chain kinase, I'm going to draw this little mechanism right here, means we are going to add a phosphate to our myosin head, which leads to contraction. Okay. And then we also know that we use the myosin light chain phosphatase. It's going to help stop or relax that contraction. So then we'll have our myosin with that phosphate group removed from the myosin light chain. Okay, so that'll be relaxation. We'll put in parentheses over here through that enzyme. All right. Now, we also know that over here we're going to have, smooth muscle cells, so not a great sarcoplasmic reticulum. All right, so that's going to be our sarcoplasmic reticulum. And we'll know we'll get calcium, a little bit of calcium from that as well to lead to a contraction. Let's start by looking at the effects of parasympathetic influence on the contractile activity of the smooth muscle cell. So I'm going to draw this in green so we can keep it separate from our sympathetic innervation effects. So we'll start here. We're going to start with a specific muscarinic receptor, and I'll just draw this here. And this is going to be the M3, like muscarinic 3. So that's a muscarinic receptor. And what that does is it activates, well, upon acetylcholine binding, I should say, that's going to activate a G protein. That G protein goes on and binds to phospholipase C. I'm just going to do PLC. Okay, so you can think back to the first week of the block last semester. And that phospholipase, remember the phospholipase, that means we're going to split a lipid. And we're actually going to split the PIP2. So associated with this membrane here, we have PIP2, that's embedded within the membrane, and it's going to split those into two different counterparts. So we're going to get DAG, which we're not too concerned about at this point, but then it's also going to split it into IP3. You'll probably remember that one. Well, IP3, this is what actually goes up to the sarcoplasmic reticulum and binds a channel there, the ranitidine receptor, and allows for more calcium to exit that sarcoplasmic reticulum, which of course is going to lead to increased contraction. We're going to bind more calmodulin, activate more myosin light chain kinase. Okay. Now let's look at one other muscarinic receptor. Let's say over here we'll have an M2 receptor. So acetylcholine will bind to that receptor. And this is actually going to activate a cation channel within the membrane. And that's going to allow for an influx of cations, specifically here, we're talking about sodium. So we see this influx, oops, a little lag on my computer, it's heating up. Influx of positive charge for depolarization, and that further activates this voltage-gated calcium channel, right? It's voltage-gated, so we get that depolarization, so we get more calcium, which again leads to more activation of myosin light chain kinase and more contraction. Okay, so we get calcium moving from extracellular concentration or extracellular into that cell. We'll finish up then by looking at sympathetic innervation effects on contractile activity via norepinephrine. So down here, I am going to draw an alpha-2 receptor. So norepinephrine will come along and bind that alpha-2 receptor. Adrenergic receptor, and that's going to activate adenylyl cyclase. So then with some ATP input, that adenylyl cyclase, cyclic adenosine monophosphate, okay? And that is going to activate PKA, protein kinase A. And as kinases do, they phosphorylate. So via different phosphorylation events, we're going to go on and have some widespread effects. And the first of those is going to be, we're going to activate the circopump on our sarcoplasmic reticulum. So that's going to help bring calcium back into the sarcoplasmic reticulum. Now because of that, there's less calcium around, so we see less contraction. Same time here, this phosphorylation event, one of the things it's going to do is it activates this myosin light chain phosphatase. Okay? So we're going to remove the phosphate, which leads to relaxation. Remove the phosphate from that myosin head. Okay? We're also going to go up and via phosphorylation events, we reduce and decrease the amount of myosin light chain kinase, so we see less phosphorylation of the myosin head. And then the ultimate last effect here is we're going to go up and we inhibit the voltage-gated calcium channels. So that also decreases our incoming calcium. All right. So again, that leads to decreased contractile activity. So we see then, you got to think about rest and digest versus fight and flight. So with a sympathetic, that's fight or flight, right? The last thing we're going to be doing is working on digesting our food. Maybe this is a cell of your intestine, okay? This intestinal smooth muscle cell. Okay? So with the effect of norepinephrine, we're going to decrease contractions of that smooth muscle. Whereas with parasympathetic activity via acetylcholine, we're going to increase contractions of that smooth muscle cell.